I'm Daniel Levine, and this is the Bio Report. One reason that CAR-T therapies haven't been more effective at treating solid tumors is their reliance on targeting antigens on the surface of cells, which can often be present on healthy cells as well. Affini-T Therapeutics is addressing this weakness in current cell therapies by using its platform technology to develop T-cell receptor engineered T cells. Its so-called TCR-T cells are able to recognize intracellular targets and attack the drivers of mutations that are inaccessible to CAR-T therapies. We spoke to Jak Knowles, CEO of Affini-T Therapeutics, about the company's TCR-T cell therapies, how they're engineered, and why they have the potential to be effective at treating solid tumors that have evaded the power of CAR-T therapies. Jak, thanks for joining us.
Thank you, Danny, for having me.
We're going to talk about TCR therapies, Affini-T, and its platform for next-generation cell therapies. Before we get into the technology, though, I'd like to start with the founder of the company. It seems there were a lot of relationships where the pieces came together nicely here. Can you just explain how it came about?
Yeah, happy to. It's a really cool story. So my background, I'm an MD by training, but I've been working for about the past 4 or 5 years with different venture groups, one of which was Bayer. They just formed a new venture fund called Leaps. And so I was managing the Leaps portfolio here in Cambridge, Massachusetts, and was lucky enough to be introduced to a number of different academic institutions, but one of them was the Fred Hutch. And so I had actually looked at starting a different company with the Fred Hutch about a year prior to launching Affini-T. And I kept in good contact with them. And they actually made the introduction to Dr. Phil Greenberg. Most people know Phil from his work in immunology. He's one of the founders of the immuno-oncology space, but also through his work with a company called Juno Therapeutics. So Juno was working on one of the first CD19 CAR-T programs, very successfully, was able to develop a drug called Breziani for that target. And also the company was acquired by Celgene, I think for around $9 billion. So Phil was really well known. I had never met him before, but was lucky enough to earn his trust. And during the pandemic, right in the beginning, we launched the company, actually. I'd never met Phil, so it took about 9 months before I got to meet him face to face, but really just formed a great relationship with him over Zoom and was able to inspire him to recruit a few of his colleagues from the Hutch, Aude Chapuis and Tom Schmidt, to join the effort as well. In the early days, it was actually me that was funding it. Bayer would not. They let me start the company, but they wouldn't fund the company until I left. And so our seed investors were myself, and then some of our SAB members, Jim Allison and Pam Sharma, who know Phil for many, many years and are big believers in immuno-oncology as well. So they were some of our first investors and also joined our scientific advisory board. And then finally, our lead investor, obviously, Bayer Leaps did come in as a lead, but our co-lead was Vita Ventures. And so I've known Arjun Goyal and Stefan Viktorovic there for close to a dozen years. And of course, the other co-founder of Vita is Ari Beldegren, who was the CEO of Kite Therapeutics. So many of your listeners remember Kite and Juno were the two companies kind of in this race to see who would be first for a CD19 CAR-T therapy. So we're very lucky to have incorporated all these different elements. We've got, Vita representing Kite, Phil representing Juno, and then more recently have hired two great individuals. One, Dirk Nagerson, our chief medical officer, who was at Amgen working on KRAS drugs, the first KRAS drug, Lumakras, and Dan Fega, who serves on our board as an independent director. He is currently CEO of Anapsys, but previously COO of Mirati, the other small molecule KRAS company in this space. So a lot of great people, and lucky enough to bring them all together to work together on Affini-T.
CAR T therapies have been effective in treating liquid tumors, but solid tumors have been much more difficult to treat. Why are solid tumors harder to treat with CAR Ts? What's the challenge?
Yeah, it's a really interesting question, and I think it's puzzled a lot of people. When Kite and Geno were acquired, I think the idea was that they were going to go on and solve lots of different cancer indications, not just hematological malignancies. The issue is really several factors. The one that we always talk about here at Affini-T is that targets really do matter. And for the heme CAR T programs going after CD19 and BCMA, those are great targets because they're on 100% of B cell tumors. They're also on 100% of healthy B cells. But the reality is patients can live without their B cells. You can eradicate the entire B cell population of an individual and, make them prone to certain types of infections, but they actually can lead a normal, healthy life and go on really to enjoy, their life till the end. That's not true for other targets. So for most solid tumors, there is no magic target that's only expressed on the solid tumor. Most of those targets are also expressed on healthy tissue. And so when you try to go and attack those tumors with a CAR T, you're also going to hit a lot of healthy tissue, and the result is unfortunately a lot of toxicity for some of the targets for solid tumor CAR T therapy. TCRs are a little bit different. A CAR is essentially an antibody, an FCFE region from an antibody, conjugated to a co-stimulatory signal that activates the T cell to kill. So if the CAR T sees that signal, it's going to kill the cell that it's engaged with. TCRs work a little bit differently. It's kind of working with the way nature designed, or I guess evolved, an immune system. The TCR is recognizing intracellular targets. So these are mutations for cancer, or you may recall for COVID-19, everyone was talking about T cell immunity. So what did that mean? It meant you had a population of T cells that could recognize COVID-19 protein being expressed on the cell surface. Your immune system is really good at recognizing foreign antigens. And so COVID-19, or in this case, mutant KRAS, are both proteins that don't exist on healthy tissues. And so T cells, if they have the right TCR, are very capable and very good at being able to eradicate those cells. So what we're doing here with TCR therapy is giving our cells an opportunity to recognize those internal mutations. And there so far isn't a great way to do that with CAR T.
The antibody part of the therapy is being used here, I suspect, as a targeting mechanism, but does it have any therapeutic benefit beyond that?
That's right. So instead of using an antibody, we're using a TCR, T cell receptor. So it's a little bit different than an antibody. It's a lot more sensitive. So you can detect very, very low amounts of mutant KRAS in this instance. By itself, a TCR is just a binder. So it's targeting the mutant protein, but it's also targeting something called class 1 or MHC class 1. This is the cellular machinery that presents foreign or even self antigens on the cell surface, allowing your immune system to recognize like a self from non-self. So for a TCR directed against mutant KRAS, by itself, it would just bind that mutation. It wouldn't actually do anything. But if it is part of a T cell, then it's on the surface of a very potent killer for your immune system. And so it's really that combination of having a TCR on the T cell surface that makes our product work. You could use TCRs in another way. Some companies are creating what are called bispecifics. So you have the TCR engager part, and then you have a CD3 arm that can also bind to T cells. And so instead of having to infuse a cell therapy, you can just infuse the bispecific, and that can both target the cancer cell and recruit a healthy T cell to then complete the tumor cell killing.
Affini-T has developed a 3-part platform technology. Walk us through each of those parts, starting with how you go about choosing a target?
Yeah, no. So our three platforms are Tailor, Tune, and Thrive. The Tailor platform incorporates machine learning, some algorithms, predictive algorithms, and artificial intelligence. And what we're doing here is really identifying the right epitope initially. And so we're going after what are called shared or public neoantigens. So these are the same, 9 or 10 amino acid sequences that are presented across the entire population. So very important to go after these shared neoantigens because you can address the most patients possible. We use the machine learning to then also predict what the best TCR will look like. We are sourcing our TCRs from either healthy donors or from patients. So these TCRs all have been in at least one person before, which is an important safety feature for us. But more importantly, you can find very high affinity, naturally occurring TCRs, but you have to look very, very carefully. When we sourced our first TCR for a G12V, we had to go through 15 to 20 donors. Each healthy donor has about 200 million TCRs in their peripheral blood. So really, it's 3 to 4 billion TCRs we're sorting through to identify one that is optimal for therapeutic development. The second platform we have is, and I should actually add, in the Tailor platform, Affini-T is really just the beginning, right? We're looking for something that, recognizes the mutation and can bind very well. But the big thing we want the therapy to do is obviously kill cancer. And so, we screen on Affini-T. We obviously make sure that the TCRs are safe. So we do a very comprehensive safety screen using XScan, which is the gold standard, also allogeneic screens to make sure it's not cross-reactive across other wild-type proteins. But the final step is really avidity, making sure the TCR-T cells actually kill. And so our Tailor platform is really optimized, one, to pick high-affinity TCRs, but really to pick the ones that actually kill cancer the best. The second platform we have is called Tune. Tune is a synthetic biology platform. And so here we're optimizing the cells not just to survive, but to really overcome the tumor microenvironment. And the first technology we're using converts a fast ligand death signal. This is the dominant way that T cells are deleted in the tumor microenvironment, and converts that into a 4-1BB co-stim signal. So going back to the CAR T example, all CAR T uses both a antibody as a binder, but then a co-stim. So many of them either use 4-1BB or CD28 as the co-stim. We're, I think, one of the first TCR companies to take that learning and apply it to TCR-T cells. So we're incorporating this co-stim signal delivered by a fast 4-1BB switch receptor. We have several other synthetic biology components, but I'll pause there and happy to answer any questions you have about the Tune platform.
Well, let me ask you about the synthetic biology aspect of this. What are you actually engineering cells to do? How much functionality can you put in there?
Yeah, so this is what's been, I think, a great explosion in the cell therapy space recently. People are using different kinds of logic gates. I worked with a few of these when I was at Bayer. They're using different synthetic biology to reprogram cells. What our co-founder, Phil Greenberg, hit upon is that, it's hard to change something in a cell without having a downstream consequence. And that's because, biology is in this delicate balance. And so if you knock out a gene or if you get a gene to constitutively express, there's frequently a trade-off. The cell doesn't become as effective at doing what it's supposed to do, which in this case is kill cancer cells. What Phil aligned on was, a very elegant approach of, instead of, trying to force the cell to do something it's not comfortable with, kind of using a judo-like approach of turning the tumor's biology against it. So the tumor is expressing fast ligand, which normally would couple to the fast ligand, so the fast receptor on T cells, and push the T cells towards apoptosis. So instead of that inhibitory death signal, we're now converting the fast ligand signal that the tumor is trying to give to the T cell to kill it to a 4-1BB co-stim. So that should only happen in the tumor microenvironment. We're not expecting the cells to get this signal anywhere besides the TME. And because now, instead of a death signal, it's getting a proliferation signal, instead of the tumor deleting the T cells, it's actually going to, encourage the T cells to proliferate and expand, making more of themselves. So it's a very elegant way of saying we don't have to constitutively express a 4-1BB co-stim, it's only going to be active where we want it, which is in the tumor microenvironment. So I kind of like this switch receptor approach because it doesn't require the cell to do anything that's going to be different than what it would normally do. It's just giving it an opportunity, one, to not be deleted by fast ligand, but two, to get a co-stim right where you want it in the tumor bed.
Affini-T's 2 lead programs target KRAS mutations. This is AFNT111 and AFNT211. What are the initial indications you're pursuing?
Yeah, so KRAS is the most dominant oncogene driver mutation. So it's very important for tumor biology. It allows the cells to proliferate and expand. And it's kind of like a cheat code for cancer cells. Without it, they don't function as well. And in fact, others have talked about this. The cells appear to be almost addicted to this mutation. So once they acquire the KRAS mutation, they can't lose it, and they kind of need it to survive. What's great about our Affini-T111 and 211 programs is that they're both targeting the same mutation, KRAS G12V. The difference is Affini-T111 is unarmored. Affini-T211 has that fast 4-1BB synthetic switch we just talked about. Both of those programs are targeting the same patient populations. And really here, we're just trying to go after the most unmet medical need and help the most patients possible. So the types of tumors that have most frequently KRAS mutations are, pancreatic, colorectal, and lung cancer. So our phase 1 study is going to be a basket trial that seeks to recruit patients from all 3 of those tumor types. We're also going to have a 4th arm that, is kind of a take-all-comer opportunity. There are some other tumor types, endometrial cancer comes to mind, where KRAS is also pretty common. But really, we want to focus on the big 3. There's a huge unmet medical need in pancreatic and colorectal cancer. Almost no drugs have been approved for those indications over the past 50 years. And most patients are still getting, chemo and radiation as first-line therapy. If they fail that, they're offered palliative chemo and radiation. So, really toxic therapies that most patients don't tolerate well. And there's been really no innovation for these patients for the past 50 or 60 years.
And in terms of patient selection for the trials, are you using any kind of a companion diagnostic?
Yeah, so we do, of course, have to screen for the KRAS mutation. So there are lots of different mutations possible in KRAS. We're going after ones on the G12 codon, and specifically a point mutation. So we're focused on G12V, which stands for valine, which is an amino acid, and G12D, which stands for aspartic acid. And so those are the 2 most common KRAS mutations. They represent about 60 to 70% of all mutant KRAS isoforms. And so that's really the rationale for focusing on those initially. We're trying to help the most people possible.
What's known about either of these experimental therapies from the preclinical studies?
So what we know is that, one, in our hands, our TCRs work very, very well. We've presented data at AACR just recently in collaboration with Chris Klebanoff from Memorial Sloan Kettering. He's our newest co-founder. He just joined for our G12D program. That's the Affini-T212 program. For Affini-T111 and 211, we've presented at SITC last year, at ASGCT, and really phenomenal data. A single infusion of Affini-T111 or 211 is sufficient to provide great tumor control, long-term tumor control across many tumor types. We've tested this in vivo models for pancreatic, for colorectal, for breast cancer, and it seems to work across all tumor populations. We've tested it in vivo. I will add there's also some clinical precedent here. So not anyone affiliated with our company, but another very prominent researcher, Eric Tran, who used to work with Steve Rosenberg at NCI, he was able to isolate a KRAS positive T cell in a patient with pancreatic cancer. He then took that TCR, engineered it into a new cell therapy for a second patient, and that therapy provided great tumor control, I think shrinking the patient's lung metastasis by 72%. So we have great preclinical data for our programs, but there is already clinical precedent and proof of concept out here that a KRAS TCR can work for solid tumors in patients.
What's the development path forward?
So right now, we're really focused on getting our programs into the clinic. Affini-T111 is targeted for IND submission in August of this year. That's a study that will be open and enrolling at the Fred Hutch Cancer Institute in Seattle, Washington. And Affini-T that's the company-sponsored program. So that IND is slated for October of this year. Here, we're going to have a multi-centric study from day one. So we're going to open enrollment at 10 Tier 1 cancer institutions in the United States. So we'll have representative geographies on the East Coast, the West Coast, Middle America, and obviously in the South. And the goal here is to recruit 10 to 15 patients into each study. So hopefully we're recruiting somewhere between 30 and 40 patients over the next 2 years.
Given the broad potential for the approach, how do you go about prioritizing targets and indications?
Really, it's about following the science. So we look for targets where our TCRs appear to work really, really well and across all cell lines. Frequently you'll see people make something work once or twice, and then they move forward with it. We've been very selective. We only want to work with cell therapies that work in all tumors tested. And so far, we've been very lucky to achieve that with both, KRAS, G12V, and G12D. In terms of the indications, we're really trying to help the most people possible. So we're prioritizing those populations where there is a huge unmet medical need in areas like pancreatic and colorectal. And then in lung cancer, just looking at patient numbers. Where can we help the most people? That's where we want to focus our attention.
Affinity is focused on cancer, but beyond cancer, what's the potential for this technology, and would you explore other potential applications?
Yeah, it's a great question, Danny. So I didn't share my background with you. I'm actually an MD by training, and spent time early in my career thinking I would be a head and neck surgical oncologist. And so oncology is near and dear to my heart, one, because it's affected so many people in my life and in my family, but two, because it's where I was trained initially as a physician. I believe very strongly that it's important to focus. And in this environment, I think, the companies that are being rewarded are the ones that are really focused on getting products into the clinic and really doing something meaningful for patient lives. There are potential use cases for this type of TCR technology outside of the oncology space. I think we just saw today, T-Scan and Amgen announced a collaboration looking at TCRs for autoimmune disease. I think that's great, and I applaud anyone who can, can focus on oncology and non-oncology. My strong belief is, patients really want these therapies now. And for a lot of patients that have pancreatic and colorectal cancer, they don't have options and they don't have a lot of time. So we're really focused on making sure we get these programs into the clinic and can help cancer patients quickly. I'll leave it to others to solve how this technology could be used outside of oncology.
You talked a little about how the company came together and the connections between you and your team and your lead investors. It's a difficult financing environment. I'm wondering, financing here was a bit different because of these relationships, but what is the discussion like today with other investors, and having sat on both sides of the table, how do you look at the current financing environment?
Yeah, financing is a challenge. What I will say is, in my experience, the good companies are always able to raise money. We're very lucky to have great, lead investors in Vita Ventures and Leaps by Bayer, but really strong investors across, all of our Series A and B syndicate. Catalio Capital, Pisces, Sean Parker's foundation, Agent Capital, Alexandria, Pinnacle, to name a few. These are groups that are really committed to the long-term vision of getting drugs approved. So we've been very fortunate to have such a strong syndicate. And so far, they've been very supportive, obviously, financially, but also, cheering us on to get our programs into the clinic. In terms of the financing environment, I think it's tough. I think, early stage companies, Series A companies seem to still be getting financing. I think all the venture funds raised a lot of capital, so they have to deploy it in order to continue collecting management fees. And And then I think, the clinical stage companies, we just saw Accelerin flip public earlier this week. They raised $621 million as a phase 3 company. So I think, the companies that have, real clinical data, strong programs, they're not having a problem raising money. And the early stage companies, because venture needs to put their money to work, are also not having that much difficulty. Where there is a lot of difficulty is that valley of death, right? Series B to crossover. We're lucky enough to have our Series B syndicate already baked in from our initial financing, but I've seen a lot of companies struggle raising money for their Series B. I expect we'll see a lot of companies start to falter if they're unable to bring in new investors. Crossover investors, likewise, are still very focused on the public markets. They're still seeing a lot of value in publicly traded stocks. Things are really priced low, so it's very attractive. And then public markets have the benefit where they can get liquid very quickly. They're not locked up in a private company for months or years. They can buy the stock one day and sell it the next. And so I think the crossover investors are still mostly focused on public markets right now. But I expect with the success of the Accelerin IPO, they traded up over 30% this week. I think we'll start to see hopefully a little bit more of an opportunity for companies to go public. And usually that starts to attract the crossovers as long as they're making money.
Given the current financing environment, do you find you're thinking any differently about your development path and how you're prioritizing the projects that are in the pipeline?
Yeah, it's an interesting question. I think everyone, should always strive to be capital efficient. And that just goes back to my venture capital training. I would always say, it doesn't matter the market. You should always be thoughtful about how you're spending your money. I think we will focus, obviously, on clinical development going forward. We have, now 3 great programs, Affinity-111 and 211 targeting KRAS G12V, and Affinity-212 targeting KRAS G12D. We plan to have a 3rd target, 4th IND for P53 at the end of next year. And I think once we have those 3 programs in the clinic, that's really where I want to focus our capital. Most of the value that's going to be created is in therapeutic development. So preclinical R&D is important. It's certainly key to what we're doing here at Affinity. But I'd like to see most of our proceeds being focused on getting clinical data. I think that's really the lifeblood of any biotech company.
Jak Knowles, CEO of Affini-T Therapeutics. Jak, thanks so much for your time today.
Danny, thank you so much. This was a lot of fun.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.